The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and welcome to this afternoon's webinar. This is Paige Nuez. I'm with RettSyndrome.org, and on behalf of our Board of Directors and team, we're so pleased to bring you the session today on clinical trials in Rett syndrome and an update from a panel of speakers. Before we get started, I want to do a quick orientation about how GoToWebinar works for those who are joining us for the first time today. For those of you who've been with us all week, I hope you've enjoyed the webinars, and we're so happy to have you with us. We're so happy to have you committed to your children, and being with us today really demonstrates your eagerness to hear about research, and having you with us all week, your eagerness to learn about how the best care practices in Rett syndrome. So for the orientation about GoToWebinar, if this is your first time, I want to let you know that the audio feed is open only for our presenters, so you don't have to worry about any background noise at your home or office, wherever you are, interfering with the presentation. We are recording today's presentation, so you also don't have to worry about taking notes. At the conclusion of our virtual conference, we will send email links to everyone attending so you can revisit the information. If you look in the upper right corner of your screen, you'll see a GoToWebinar control panel, and in that control panel is a link where you can type in questions for our presenters, and we really encourage you to submit any questions that you have, and we'll reserve time at the end of today's talk to address as many of your questions as we have time for. If we don't get to them all today, we'll circulate a question and answer document to our panelists and get answers out to you by email shortly after we conclude our virtual conference. Okay, if you're having any trouble at all with the audio, check the volume control on your phone or your device and make sure speakers are connected and everything is turned on. As we go through the presentation, if you have any trouble seeing the slides or hearing the audio, sometimes just exiting out of the application and then logging back in clears it up very quickly. But again, just let us know in the question box if you're having any trouble, and Megan or I will help you troubleshoot that. So that we can get to all of our important content today, I don't want to spend too much time on introductions. We'll have every presenter introduce themselves when they get started. We're going to start today with Steve Kaminsky, who's Chief Science Officer, RettSyndrome.org. And Steve is going to give us an overview of full spectrum research strategy for RettSyndrome.org, and then we're going to hear from some of the clinicians who are running clinical trials and the natural history study. So Megan is going to turn the slides over to Steve right now, and we can get going. While we're getting that up into the slideshow view, just remember you can submit any questions for any of our panelists through the question box, and we'll field them at the end. So I'm going to...
I want to this is Steve Kaminsky, the Chief Science Officer of RettSyndrome.org. I want to make sure that everybody can see my first slide, which says Virtual Conference with RettSyndrome.org Research Agenda. Megan, Paige, can you see that?
Yes, we can, Steve.
Okay, so I'm going to get started. So you've all been hearing all week long about the research that's been going on in Rett syndrome, everything from basic science up through including the natural history study. I'm going to give you our research agenda today, and what I hope to do in a very short 6-slide presentation is to take you through what we're trying to do at RettSyndrome.org as it stands today. So on this slide, you can see on the left-hand panel is that RettSyndrome.org has invested heavily into basic science. And in that basic science, we've done things from the standpoint of an association of you know, what is MECP2 doing in Rett syndrome, what are the functions of MECP2, the biochemical pathways, when there can be genetic reversals, how is it done, what does that mean? And then in the animal models, what does pharmacologic reversal do? How is it done? What compounds are used? And then most importantly, it's trying to understand how do you activate cells in Rett patients, and how do you activate and understand networks. So to this end, we've actually invested, since the beginning of Rett Syndrome.org, a lot of money into the basic research to really put down a foundation of understanding. And today we are still investing in that. And that's because we're still unraveling all the mysteries of MECP2 and what it's doing both developmentally, not only in the central nervous system, but throughout the body. So we continue to invest in the basic science. Now, at the same time, 12 years ago, a natural history study was started, and you're going to hear a little bit about that from Dr. Percy following me. But that natural history study is incredibly important for all that we're doing at RettSyndrome.org, because what we're doing is assembling how many people are affected, what are the actual variants, and what do those variant forms of Rett syndrome look like, what's the standard of care for each of the girls, and what's the best care for each of the girls. How do we move towards earlier diagnosis, trying to find essentially outcome measures that can be used in future clinical trials. And lastly, and one of the most recent things we've been trying to do is build a patient or contract registry, all of which will be really important as we move forward with studying Rett syndrome. So this basic science that we've been really focused on really has a couple of things associated with it and why we're actually taking these approaches. One, by better understanding the gene itself, it would give us better hold on how we might fix the gene, what modifiers might be out there, because with each of the girls with the same mutation, they might have very different profiles in regard to Rett syndrome. And lastly, by understanding the biochemical pathways that are associated, we might be able to essentially look at those pathways and understand which of those pathways we can actually study to essentially try to do therapeutics. Now, at the same time, so that's sort of approaches to correct the biology of Rett syndrome. At the same time, one of the things we're doing is how do we approach Rett syndrome from the standpoint of essentially resetting neurology. And this is really important because this is a neurologic disease, and our girls and boys and adults with Rett syndrome don't have all the requisite networks to actually undertake all the things that you and I take for granted on a daily basis. So our program is designed to actually try to understand how can we stimulate synaptogenesis, how can we enhance neuroplasticity, and neuroplasticity is how do we get certain parts of the brain to do things that they don't normally do, but in fact they can rescue some of the networks that we're hoping to stimulate. And we can do this through building sort of rehabilitation or habilitation protocols through advanced physical therapy or occupational therapy, and hopefully speech therapy, and hopefully cognitive therapy. All of those approaches to reset neurology. Now, you've heard this week that, in fact, there's a bunch of non-compound or non-drug clinical approaches that we're really going after. And this is part of that, because if we can stimulate synaptogenesis and enhance neuroplasticity, even without drug intervention, that will actually change the quality of life for all of our patients, boys, girls, and adult women with Rett syndrome. So all of these essentially lead us to one other thing, and that is really rigorous preclinical trials. And you've been reading about some of our clinical trials, and both Dr. Percy and Dr. Kaufman, who follows Dr. Percy, are going to be talking about some of the clinical trials that RettSyndrome.org are associated with. But why are we doing these? So these rigorous preclinical trials you heard about earlier this week from John McCall and Danny Bruner about screening repurposed compounds, looking for new compounds, all of which go back to how do we stimulate downstream pathways in Rett syndrome. And in so doing, what that will do is give us possible ways to essentially approach Rett syndrome from a clinical trial standpoint. And the other piece is this whole the whole idea of non-drug clinical intervention, and that is look for novel ways to essentially help with gain of function. And those really are research. It's doing the kind of physical therapy research, occupational therapy research that will take us to the next level with children and adults with Rett syndrome. So all of these rigorous preclinical trials are really geared toward bringing forward more clinical trials. And what are we trying to do with these clinical trials? What we're trying to do with these clinical trials, as you all know, one of the challenges with Rett is that we don't have enough synaptic connections. So some of the drugs that we're using in our clinical trials are just that, to enhance synaptic networks. If we can enhance synaptic networks, then our next challenge is how do we wire and fire those networks? And that's why all of the approaches to reset neurology are so important to us. So that whole idea that Rett Syndrome.org has is the idea of, one, correcting the biology with drug therapeutics, and two, then resetting the neurology with ways to fire and wire networks. Now, this is essentially what I call a full spectrum approach to Rett syndrome. And our hope at RettSyndrome.org is that this two-pronged approach, correct the biology, reset the neurology, is going to help change essentially the quality of life for the girls now, as opposed to waiting for the magic bullet 10 years or 15 years down the road. We still want the magic bullet, and by God, we're going to get there. But until we get there, we have to do the best with the compounds we have, and both the physical and the occupational, the speech and the cognitive therapies to enhance all the networks that we can get to grow. Now, I'm going to stop there, and I will take a few questions because I don't want to take time from Alan and or Walter. So if there's questions, please feel free to type them in. Paige will essentially get those to me.
Sure will. Thank you, Steve. We do already have a couple of questions that have come in, especially right now, if you can talk about any research or trials that might involve boys with MECP2 mutations, MECP2 duplication in that area. What are we doing in that area?
Yeah, so that's probably a question that's best for Walter or for Alan, but let me take a stab at it. The challenge is with boys, there's such a rare occurrence to essentially do a clinical trial which is only in boys. The inclusion criteria would bring in so few boys that we would have a very hard time getting the power that's needed in those experiments, those clinical trials, to sit in front of the FDA and go forward. So our hope is to essentially take what if we're able to be successful, and when we're successful in girls and women with Rett, we can go back to the FDA for compassionate use for the boys. But the challenge is numbers. And I think that there are so few boys that we would actually be hard pressed to be able to power any clinical trial that would satisfy the Food and Drug Administration. So I think that right now it's unlikely because of the numbers. The numbers essentially are everything to the FDA. That's what they do, and they will only go, they're very conservative in that regard, and they will not put anybody at risk. And if we can't essentially power our experiments the way they need to be powered, they won't let us go forward. And I would ask Walter and Alan to add to that when they are speaking. That's a good question, and it's one that it deserves a good answer. So Walter and Alan, if you could write that down and essentially address that as well. That's my opinion, and I think that, in fact, that's probably, what's driving the day right now. Other questions, Paige?
Great. Thank you, Steve. And I will ask that again when we open for questions at the end for Dr. Percy or Dr. Kaufman. Okay, so Megan, if you'd like to go ahead and let Dr. Percy pull up his first slide, we can kind of take care of those logistics while we ask the next question. Steve, one of our parents asked, where can they find all of the research that RettSyndrome.org is involved with on our website? Where on our website would you point families to?
Yeah, so if you go to the research section, actually all the research we're funding is actually in that section. If someone can't find it, please feel free to email myself or Janet, Dr. Janet Toscano. We can actually help provide that. But Janet has essentially set pages of that, what we funded in the past and where it's being done. So it's on our website under the research tab, and then when you pull down the research tab, you'll see currently funded programs.
Great, thank you. That's a great question. And oops, I just hit reply. I'll send the link out to everybody and include your email address in the chat box. I'll also point families to, there's an area on our website. If you go to rettsyndrome.org under For Families, you'll see a section called Contributing to Research. So if you want to look at all of the research being done in all of the quadrants that Steve talked about, you can find that under the research section. If you'd like to zero in on areas of research that you specifically as a family can contribute to, whether that's a database, a natural history study, a clinical trial, a non-drug clinical trial, donating tissue samples for scientists, those are all listed under the For Families section, Contributing to Research on our website. Thank you, that was a great question. Okay, so in the interest of time, if we can, let's go ahead and have Dr. Percy start up with his talk, and we'll come back for questions a little bit later in the presentation. Thank you, Steve.
I hope I'm up. And good afternoon, everyone. Hope you've had a good week, and both entertaining and educational. And we will hope to continue this with the discussion of the ongoing clinical trials. My task is to discuss the NNZ-2566 trial, which was completed about 9 months ago in girls, older girls and women. And we've recently been at the FDA for a discussion of the further trials in younger girls, as well as the previously studied young adults and adults with Rett syndrome. So just to touch briefly on the natural history and to reiterate why it is so important for natural history data to be available to help guide and instruct us in the ongoing clinical trials. It is important to know how a disorder evolves naturally, so that when you institute a trial, whether it's a medication or a device or a therapy, that you can demonstrate definite improvement, and better yet, perhaps, you can know if the trial is actually causing some harm, and we certainly do not want to do that. So the natural history study, which we wrote for approval and was approved in 2003, had the initial goal to enroll 1,000 girls or women with Rett syndrome. They had to meet criteria or have MECP2 mutations, and the purpose was to expand the phenotype-genotype relationship that exists in girls with Rett syndrome, and then to set the stage for clinical trials. Initially, through the first phases of the study, first Baylor, Greenwood, and UAB were involved, and then with the second iteration of the protocol, the site at Boston Children's Hospital was added. Now, you can see from the geographic representation of the principal sites that we didn't cover the U.S. very well, and therefore it represented a potential barrier to parents and other family members to actually enroll their daughters in this study. Therefore, we created a system of travel clinics where we went to these 4 sites twice a year on weekends. So we spent 8 weekends a year from 2006 until middle of 2014 doing these travel clinics. They were very important because they added about 40% of our total visits. All of the data that we acquire in these visits, whether they're at home or on the road, were uploaded to the contact registry, which is at the Data Management and Coordinating Center at the University of South Florida. That is a website which you can see there. I would encourage everyone to have a look at it. There is both a public portion to that website as well as a members-only portion. The public portion is can be very instructive, can tell you what is going on in this disorder as well as other disorders, and can also tell you how you could be further involved with studies. One particular note that I would make, and to emphasize what Steve said, is the establishment of a contact registry. Now, the contact registry has been available through the DMCC since the onset of this study, but it has not really been utilized well by our consortium, or certainly for not for Rett syndrome. It was for the other disorders which were involved. So in the first two iterations, our study involved both Rett syndrome as well as Prader-Willi and Angelman syndromes. So the contact registry it allows individuals voluntarily to enter information, and if they want to give further permission, they can agree to be recontacted if new information arises or new questions arise that we might want to address to the members of the contact registry for potential answers. The current enrollment is actually now more than 1,205. It's 1,208 or 10, but these participants are listed below with their breakdown. As I said, 40% or so have been enrolled through the travel clinics, and so we have those with Rett syndrome are 940, those with variant forms of Rett syndrome 166, and lo and behold, we came up with 99 individuals who have alterations in the MECP2 gene but do not have Rett syndrome. This was not something we really anticipated early on, because both MECP duplications and the identification of mutations in boys who might or generally do not have Rett syndrome were really not known. But since that time, we have identified 51 females and 48 males who have such mutations and yet do not have Rett syndrome. Beginning in August, actually it was in September, but the funding year essentially goes through the end of July, so that the second year would start August 1st. We had a new study funded, and this study, because of the knowledge gained with MECP2 duplication and with other Rett-related disorders, not only those individuals who have mutations in MECP2 but do not have Rett syndrome, but also individuals with mutations in the two other genes, CDKL5 and FOXG1, and perhaps other mutations may arise in the future. So this new natural history study deals only with Rett syndrome and its related partners. We are due to begin this July with a new protocol, which is much expanded. In fact, it has 3 separate protocols, and I'll jump down to the bottom to describe those. One is the same natural history study, but an expanded version of it with some additional questions. A study of the biomarkers. So we know, as Steve pointed out, that girls with the same mutation may have quite different physical and neurologic characteristics, and why would that be? And one reason could be differences in the phenomenon known as X chromosome inactivation. So we will be looking in detail at X chromosome inactivation in girls. And we will also be looking at polymorphisms in a gene, which is very important in the biochemical pathway in the brain. That is a gene, or the protein, called brain-derived neurotrophic factor, BDNF. And the third protocol will be an advanced study or observations and analysis of neurophysiology and neuroimaging. The biomarker study is led by Dr. Newell, and the neurophysiology and neuroimaging study is led by Dr. Kaufman. So the goals set for enrollment in these protocols are 1,000 girls or women with Rett syndrome, and perhaps the occasional boy, 100 generally males with MECP2 duplication, although we know that a high percentage of mothers of these boys share the same duplication. And then the Rett-related disorders, we will enroll 100 in total. The team, which is currently aligned to be involved in this study, is listed here. I'm not going to read all those names through, and you can certainly see them on the saved version of these webinars. I might point out, however, that in addition to these sites, we have 3 sites in which we're in very close and we hope final discussion about having them added as enrolling sites as well. These sites, because of the limited amount of money provided by the National Institutes of Health, these sites would be funded completely by rettsyndrome.org. The sites are at the Gillette Children's Hospital, the Cincinnati Children's Hospital, and the Washington University of St. Louis in St. Louis, Missouri. One benefit of that is the largest enrolling travel clinic that we had was in Chicago, with families coming from as far north as North Dakota, northern Minnesota and Wisconsin, and as far south as Missouri and Arkansas, and as far west as Nebraska. So it covers a broad geographic area, and these 3 sites do greatly enhance our ability to provide coverage throughout the country. With that in mind, I'm going to step over now and talk about the NNZ clinical trial. The first positive lead, as Steve implied, and there was a positive lead, and that arose in studies which were published in 2007, in which the MECP2 gene was inserted into the genome of mice who had no other MECP2 gene, but that gene was under the control of the estrogen receptor and would not ordinarily be expressed until the estrogen receptor ligand was provided. And when that was provided, whether the animals were in early pre-symptomatic or symptomatic phase, or were more mature and were desperately ill, those animals improved dramatically. They may not have improved 100%, but they improved 90%, and so we were very much taken by this as a proof of principle that if we could find a treatment which would alter the absence or abnormality of the MECP2 gene or completely replace it, we could, in fact, lead to great improvement in these individuals. So NNZ-2566, which was the name that we recognized with this compound, it has since been given the name Trofinetide, but for the future I will just say NNZ-2566 or 2566. And it is the terminal tripeptide of the IGF-1 peptide. And Dr. Kaufman will be talking later about the use of the full peptide. This tripeptide was chemically modified to be available for oral use, which is a great assistance in any trial or therapy. The IV analog of this compound had been used in traumatic brain injury among service personnel and is still being trialed by the Department of Defense or through the auspices of the Department of Defense. Safety data had been derived from the TBI study and provided the impetus and the reason that FDA approved the early phase 2 trial in individuals 16 to 45 years of age with Rett syndrome. What is the mechanism of action of 2566? While it is known and has been shown in acute brain injury, it has a neuroprotective effect in blocking a so-called form of cell death known as apoptosis. And in the mouse model for Rett syndrome, it was shown, one, to increase longevity, and to improve synaptic connections and spine densities, and overall to suggest a stabilization in cortical plasticity. I have additional information, which is that some of the other genes or proteins that are important for brain development are stabilized or brought to normal levels in the same models. So this phase 2 randomized double-blind placebo-controlled dose escalation study was what was proposed. It's very important that a double-blind study be conducted so we know exactly that the results we are seeing are objective and not based on either something that the parents feel or the physicians feel. So double-blind means that neither of us knows who is taking what medication, the real drug or the placebo. The dose escalation of this was to study a group of girls or women with a 35 milligram per kilogram per day dose, and then if that were without undue problems, to advance to a 70 milligram per kilogram dose. As is true in most early phase 2 trials, the primary outcome measure was safety and tolerability. You all know that the Food and Drug Administration is tasked with the job of making all drugs safe, and they hold very firmly to this mission. The secondary outcome measures we studied were both clinician and caregiver measures of severity and behavioral and physiologic measures, as well as pharmacokinetics, that is, how the drug was dispersed and displayed within the individuals who received actual preparation. The inclusion criteria were fairly strict. These were adolescents or adult females 16 to 45 years of age. They had to have typical Rett syndrome with a MECP2 mutation, and they had to have the clinical severity score, or the CSS score, between 10 and 36. The CSS score has been applied systematically throughout all visits in the natural history study since its initiation. The exclusion criteria were also fairly extensive. It could have previously been treated with IGF-1 itself. They had to have no evidence of liver or of diabetes or thyroid disease, and they had to have other evidence of normal organ function. They had to have They could not have prolonged QTC, that's the EKG or ECG change, in which the conduction of the heart rate is slowed abnormally, and they could not be on an liver-inducing anti-epileptic agent, either phenytoin or Tegretol. They had to have no systemic illness of any significance, and they had to be on a stable medication regimen otherwise. So the dosing schedules that were first started were 35 milligrams per kilogram twice a day for 14 days, and in the midst of that, the FDA granted approval for a study lasting 28 days. That first trial group was concluded with 28 days at 35 milligrams per kilogram twice a day, and then it was advanced through the DSMB approval, the Data Safety and Monitoring Board, who found no issues with the 35 milligram per kilogram dose, and we were allowed to study a group of girls 70 milligrams per kilogram twice a day for 28 days. The drug to placebo group ratio was 2 to 1. So essentially we had 18 girls on drug and 9 girls on placebo. Following the 26th day or so, because that was the taper off, by 28 days a post-treatment assessment was also conducted at 40 days of age, 40 days of treatment. The effectiveness measures that were utilized were the Motor Behavioral Assessment, MBA, which is also part of the natural history study, the Clinical Severity Scale, as I mentioned, an instrument known as the Clinical Global Impression, or CGI-I, improvement scale, which is modified to relate to Rett syndrome. The well-used aberrant behavior checklist, an analog scale of the top 3 concerns of the parents for each child, and those could vary from child to child, and then a modified apnea index, which was obtained by a prolonged EEG and apnea testing method. The results. The drug was very safe and well tolerated. There was no serious adverse event related to the drug, and no adverse events related at all to the time on the drug or to the different doses. Efficacy was noted at the 70 milligram per kilogram dose for the MBA, for the CGI-I, Clinical Global Impression and Improvement Scale, and the caregiver top 3 concerns. They also did a permutation test to see what the probability of a false positive, that is an unreal outcome, and that was 0.023, which is 23 possibilities out of 1,000, which is regarded as a very, very good outcome. So the conclusions: the primary outcome measure was accomplished, and the secondary outcome measure at 70 milligrams per kilogram per dose met the pre-specified criteria of supporting the clinical benefit for this agent. I must say that I, along with the other investigators in this trial, were pleasantly surprised with the positive outcome and suggest that further study is needed to support that this could be a real and treatment for Rett syndrome core features. The sponsoring company is Neuron Pharmaceuticals, a small company based now in Australia, and the participating sites were initially Dan Glaze and Jeff Newell at the Baylor College of Medicine. We entered somewhat later in 2013 here at the University of Alabama at Birmingham, and then Tim Fama and Art Beising started at the beginning of 2014 at Gillette Children's in St. Paul, Minnesota. I'd like to conclude really with remarks that I hope will lead into Walter's presentation and his thoughts on the IGF-1 trial. So, effective trials are critical. We need to have a targeted treatment option that's going to be able to be utilized at multiple sites, because doing one of these trials at any single site is quite difficult and time-consuming. We need and have well-defined populations both in this country as well as throughout the world. We know endpoints that are reasonably clear, and we know and how to evaluate them. One point of need is for more mature and more acceptable outcome measures. And among other things, Walter has been leading an effort to achieve these. I had a next slide which questions, but I'm going to hold those, or ask you to hold them until Walter's concluded. And with that, it's my pleasure to introduce Walter Kaufman, who's professor of child neurology at Boston Children's Hospital in Harvard, and who will now discuss the IGF-1 trial.
Great. Thank you. Thank you, Dr. Percy. And we're getting Dr. Kaufman's slides up on the screen right now. We have lots of questions coming into the queue, and we'll do our best to address everyone's at the end of this presentation. Thank you, and welcome, Dr. Kaufman. Okay, we can see your slides.
Some of the same themes and to make them more clear. I'm sorry?
Yeah, we can hear you okay now. Yeah, we couldn't hear you at first, but I think we can hear you okay now. Thank you.
Okay, like I said, I'm very pleased to be at this webinar, and what I'm going to try to do is to connect the two previous presentations and expand on some concepts, and as well as provide an update of the phase 2 IGF-1 trial. So I think that the only challenge I have right now is who is controlling the slides. Megan? Okay, thanks. So what I wanted to talk first is why do we need clinical trials? You have heard a lot about that clinical trials are so important for developing treatments, but why do we need them? Because it's the only way to know that a drug or any other treatment, for that matter, is effective in Rett syndrome or any other condition. And this involves both drugs that have been, or treatment have been developed specifically for Rett syndrome, as well as treatment that may be available for other conditions and now are being tested for Rett syndrome. Actually, the two trials that you will hear about today have a history, have begun to because of the development of treatment for other conditions, traumatic brain injury, growth deficiency. It's very important that, and as it had been mentioned by the previous speakers, the FDA considered very critical that the drug had to be safe. It's very obvious. Like as I have mentioned many times before, the FDA is in a very unique and I would say impossible situation, like the IRS. They can never win because they need to balance safety with efficacy. And like everyone hates taxes, but we need them in the same way we need an agency that control this balance. If they go too fast and they overlook safety, the population is at risk, and parents or other populations who are being targeted by a treatment will be unhappy. The same way, if they are too focused on safety, we are now going to see efficacy. So to reach that balance is one of the critical issues, and it's not only for the FDA, but for everyone who is developing a new treatment, including us, the people who are performing clinical trials. And why do we need the blessing of the FDA, or in Europe from the EMA, which is the FDA equivalent? Because it's the only way that the people who produce the drug and who will pay for the drug will be engaged in the process. There is no incentive for a drug company to develop a drug if at the end very few people are going to receive it. It cannot be prescribed because either it's not paid by a government, or it's not like in Europe, or it's not paid by a prescription plan here in the United States. So that's the reason why when you read about a trial, you have to ask the question, is this a registration trial? That means is this a trial that is intended to obtain FDA approval, or in Europe the EMA, and in Australia it's equivalent, or not? Because if it's not, unless the drug is very widely available, and for that reason we are only doing the trial to make sure there is a drug that is effective and safe in Rett syndrome. Other than that, there won't be incentive for producing the drug for Rett syndrome, and the supply cannot be ensured. So that's a fundamental question. The next slide. Now, you have heard a lot about how important is experimental research targeted new treatments, whether it is in vitro, laboratory type of assessment, or more commonly use of animal models like mice. Because even if it's very successful in a model of a disease, in this case Rett syndrome, there's no guarantee that that's going to be the case in humans. Unfortunately, or fortunately, we are not like mice. So that means that there is no extrapolation one-to-one from mice to humans. We need to be sure that it's working in humans, both in terms of safety and in terms of efficacy. And we have collected some experience in other neurological disorders. Neurological disorders affect adults, some of them young adults with multiple sclerosis, as well as experience in developmental disorder, particularly Down syndrome, Fragile X syndrome, and autism. And we have learned is that unfortunately, even though it may work in an animal model to cure or to ameliorate most symptoms, the reality is that most of the time this drug is only targeting, is only improving certain aspects of the disease. So I think this is an important point going forward, and I think this is something that Rett Syndrome .org has been thinking about, that even though these two drugs that we're talking today are successful, that's not going to be the end of the pathway, because most likely they are not going to be fully effective on everyone, on every symptom, and will need to develop new treatments, additional treatments. And so not everyone responds the same way to the drug, and a very important area of research in drug development in clinical trials is to know why some individuals respond better than others. Does it have to do with the way that they metabolize the drug, that the way their livers and kidneys deal with the drug, the way that the drug penetrates in the brain? There are so many factors that are important. And one important issue is, why do we need so many trials in sequence? Dr. Percy mentioned the meeting with FDA for the NC drug this week. Why do we need to go to a phase 3 trial if a phase 2 had been successful? Because it had to be replicated. That's the essence of science, that we need to make sure that this is a positive outcome. One time is not a random phenomenon, but is a consistent one. But also many times we need the phase 3 trial, the replication trial, to understand what is the best dose, or what is the range. Otherwise, if a drug is approved, the clinician will have to experiment with the patient to figure out how to administer the drug, what's the best dose, and how to do it. The next one. So here is a scheme that some of you probably have seen on the web. There are many of these versions of this to tell you the process of drug development and how long does it take from the preclinical stage, from the conception of the idea of a new drug, animal testing, all the mouse trial that Dr. Kaminsky mentioned, which are so important, So crucial. And then we go through that central portion of this cartoon, that is the different phases of trial: phase 1, phase 2, and phase 3. And each one of these has a particular goal. Phase 1 is the beginning, to demonstrate the basic, that the drug is safe. If the drug is not safe, that's the end of the road, obviously. Then the phase 2 trial, that are those that you are more familiar with. When I mentioned trial, you're thinking of that, are trials in which you were mentioning whether the drug is effective, whether the drug improves a particular symptom, manifestation of the disease. And we keep monitoring in phase 2 trial safety, side effects, because it's so important. Unfortunately, there is no drug that is completely safe. As you know, the most popular that you buy over the counter, one of them has become the prototype of the drug that almost everyone has taken, aspirin. Even the humble aspirin and the magnificent aspirin, it has side effects. Now, why do we need phase 3? Because we need to replicate, we need to confirm that the effects are positive. And if you see under these different phases of trial, you see numbers. And those numbers mean the number of subjects a typical trial involves. Now you're going to say, wow, those numbers are very large. Yes, those are the typical average numbers, and those are the numbers that are used, for instance, when we're developing a drug for diabetes, hypertension. For rare diseases, we cannot go that high because obviously there are constraints in finding enough subjects. So the numbers are not there to illustrate what we need, but the idea is that the scale or the magnitude of every trial is different. Phase 1 can be done, as Dr. Percy mentioned, as a single center, but phase 2 trials and on may require to be multi-site, simply because of the magnitude of the task, and important to make sure that there is replication across sites. So as I mentioned, the phase 3 is the replication trial, and it's the final time in which adverse events, side effects are tested. And after that, we can go to the FDA. As you see, we go at the beginning, we go at the end, and we can present the data to the FDA and to say, well, we were able to see effectiveness. This drug seemed to be good for this particular set of symptoms, and the FDA reviewed the data, is agreeable, let's say, with the results, and the drug is approved. And after that, even after that, that's not the end of the road. The FDA requires that continuous surveillance, continuous survey whether the drug is effective even after it is being prescribed. Now, all these numbers, as I mentioned before, and phases are slightly different for rare diseases. There's an entire system, I don't have time to go over that, maybe at the question period, that these things can be downscaled in terms of time and numbers when we're dealing with rare diseases. The FDA is aware of that and trying to help us to get drugs that are safe and efficacious. The next one. The next slide. Okay. So those are the concepts. And why we need them, how we go. It's a long process, and I know that many of you may be frustrated by the length of this. But we had to do it the right way. You know, Dr. Percy mentioned that there is an ideal sort of gold standard, which is a randomized, double-blind, placebo-controlled study. That means that the drug and the placebo are distributed randomly in the sample, in the so-called cohort. We don't know who is getting the drug. You don't know as parents. In the case of patients who are aware of being in a trial, they don't know. So that's the idea, because it's completely blind, objective, and whatever is found at the end is likely to be reproducible. Now, there are many ways to do this. We can have two groups, one receiving the drug, then the other receiving only the placebo. We can have different ratios, as was the case in the NZ trial. We can compensate for a parallel design, which is this one, drug versus placebo, by offering the a participant who didn't get the drug, who were randomized to placebo, to get the drug at the end as an extension. That's a so-called open label extension, which is a good way for getting additional data. It's not the primary source of analysis for the FDA, but it's a way to have more longer-term data and also to have more safety data. So what about crossover trials? The trials in which in the first half the participant can get the drug or can get the placebo, and in the second half the other one. Everyone get the drug. It sounds good. It sounds a good deal, but because of their own nature are very long. They are really long, and a lot of things can happen in the course of the process. So for that reason, and particularly for phase 3 trials, the FDA doesn't like them. We can use it in phase 2, but the ideal situation is a parallel design. Some receiving the drug only, and some receiving the placebo only. I think it's important that the drug is targeting important symptoms of the disease. This is pretty obvious, but sometimes it's not that obvious. We need to know what are we treating, and how do we know that? And this is one of the big challenges. I'm going to tell you more about that through the IGF-1 trial data. A very important point, which has been emphasized in the last few years by the FDA and other agencies, is that we don't want small incremental improvements. We want a really big one. One has an impact on quality of life. And so we don't want little changes. We want changes that really make a difference. And that's important because that puts a very high standard for whoever is developing trials and performing them. And several times during these two presentations, the issue of outcome measure have been mentioned. This is one of the most tricky things. And you will think that it's very easy to measure improvement, but to measure in a way that is unbiased is not easy. And developing outcome measure has been, to a large extent, overlooked and to some way neglected because it's difficult. And I think there is always the rush to test a drug, and we think that we have ways to measure how it's making a difference. And I mention this because if the differences, the improvement that the drug is making is big, we don't have a problem. We can measure almost in any way. But most of the time, because the emphasis of the FDA is on safety, what we need to do is to go slow and to go with lower doses. So when the doses are low, we have the potential problem that if we don't have a measure that is sensitive enough, we're going to miss the opportunity to detect a change. So it's very important to have measures that are sensitive and objective. The other thing, the last point in this slide is that we need industry. Unless we set up our own company, which I don't recommend anyone, we have been producing placebo for our own trial, and it's not a minor undertaking. We need to partner with the people who manufacture the drug. And in fact, the FDA likes to see the industry involved at least at the end of a trial, because that's the only way that they can be sure that this is going to go somewhere. Unless we do this as a research study, and all what we're doing is research, we're not looking for FDA approval, which I told you that it's very important to have FDA approval, because as clinicians, we want to have a drug that we can prescribe, that is available, like I always say, available and affordable for our patients. So for that, we need industry. The next one. Next. Okay, thanks. So having said all the things, I think that families get very excited about clinical trials, and in the excitement of having the opportunity to improve the quality of the life of their children, one thing that is forgotten: a clinical trial is a research project. Remember that there is no proof. If the drug, we knew that the drug was effective, automatically we don't need to do a clinical trial, but we don't know. So keep that in mind. It may not work. It may have side effects. So far, the two trials that we're discussing today have demonstrated safety, but it's not always the case. So clinical trials are research, a different type of research. It's a research or intervention. And as I mentioned before, it could not be only a drug. We could test a device or could test behavioral intervention or other types without drugs. So one of the problems is how we do all these things in a way that is expedited. If a clinical trial fails, it may take a couple of years to know. And this is really a challenge that the field is facing, and I'm going to discuss a little bit more in a minute. But there are ways in which we're trying to make this pathway shorter. Do we have to go all the way through 1, 2, and 3 phases? Can we make it shorter? Can we combine phase 1 and 2? And the answer is there have been many attempts, and this is an area in which we haven't been active as a community in the developmental disorder of Rett syndrome, but we should be. And I think that it's a time to go there. Oncology is leading the way with some type of trial that are called adaptive, in which instead of having the system consistent across the process, along the process, there are changes, but there are changes that are very well controlled. And we are working on thinking about those trials for the future of Rett syndrome. And there are some challenges. I have to tell you, parents want to start right away. And that may be happiness in the short term, but unhappiness in the long run. Because if you don't go to the FDA at the very beginning, and you made a mistake designing the trial, and you made a mistake in the way that you are doing the trial, by the time that you go to the FDA in the middle, or in particular you go at the end of the trial, they may tell you, we don't like this data, we don't like the way that you did the trial, you have to start all over again. And I think that that's a very disappointing experience, and also it could be quite expensive. Because clinical trials, well done, as you could imagine, are complex. They require a team and technology, and they are expensive. For that reason, they need to be well planned. One of the worst things can happen in research, and particularly in research about intervention, is that the trial was not designed properly, and we arrive to no conclusions. It's better to know that a drug is not effective, and you move on and you test another drug. There are many in the pipeline for Rett syndrome. But when you arrive to no conclusions, it's really frustrating, because you don't know if to rule that drug out or to start all over again. So it's so critical to have good design. And like I mentioned before, clinical trials are complex, and they require a team that learn an infrastructure. At the time in which I became involved in clinical trials, there were no way to learn other than being involved, learning by doing it. Now there are formal training programs in different medical institutions, so it's possible to learn about that. And there is nothing that is better than practice, but now we have a system in place in general for clinical trials that can make them much better, because there is knowledge and there is infrastructure. But no matter what, you need a big investment. The next one. We have learned from the past. I think that one of the trials in Rett syndrome didn't begin with the mouse models and a couple of years ago. They began a while ago. Unfortunately, the early trials were in general inconclusive. So there's a long history. But you know, there are some lessons from the present. As has been mentioned many times before, knowing what is the natural evolution of the disease is very important. And that also provides another comparison group, not only placebo versus drug, but also we can see what would have happened if the drug had not been there. What is the natural evolution? What happened to a girl with classic Rett syndrome between, let's say, ages of 8 and 12? What is the typical evolution of motor function? What is the typical evolution of breathing abnormalities, and so on? We can learn from the present also. We can look around. And it's very important that we don't stay isolated. What is happening in other developmental disorders? What's happening in autism? Can we apply anything from autism, from Fragile X, from Down syndrome? And as I mentioned, and it has been mentioned before, we need better and more meaningful outcome measures, particularly those that we tend to call biomarkers that are objective. And there is never enough planning. Planning, and more planning is important. We need to know everything. What type of drug are we using? Which symptom are we targeting? We need to be thoughtful about every step. And also we need to be reasonable in our expectation. And everyone has to play a part in this. From the investigators, from the advocacy groups, from the funding agencies, if there are others, industry, and so everyone has to be on the same page. They have to expect the same. One issue that has come up recently and is almost threatening some trials is the use of social media. Social media, Facebook, and other ways to communicate with each other has a positive side. We can spread the news. We can recruit participants to a trial. But if everyone is starting to talk about how the participants should be, what are the symptoms, what are the inclusion criteria, and we are beginning to use that data, we are affecting the integrity of the trial. And guess what? The FDA knows about Facebook. If you thought that they have not learned, they know about that. And some trials have been stopped simply because they have been, between quotes, contaminated by so much talking, so much influence of conversation. And at the end, it had been an unfortunate situation because trials had to start all over again. Remember, this is a partnership. I don't think there's any type of research these days that is not a partnership between investigators, the participants, in our case, the children and their parents, their families, and our advocacy groups. Everyone is involved in this, and we need each other. The next one. Next one. So I wanted to mention about something that is happening. As some of you may know, I am moving from Boston to South Carolina. So the obvious question, why and when and why? Let's start with where. I am moving to the Greenwood Genetics Center. So many of you know about the Greenwood Genetics Center. As Dr. Percy mentioned, it has been a very early participant in the National History Study. I could say, perhaps, I didn't check it with Alan if he agreed with that, but we can say the Greenwood Genetics Center is the genetics home of the National History Study at this point. So it's a very large independent center for genetics, both clinical and laboratory. And what I'm going to do there is, as part of the national evolution of the institution, they are ready for developing treatments. So I am going there to develop what we have called a Center for Neurogenetic Therapies, which is going to hopefully accelerate the development of new treatment, specifically for neurodevelopmental disorders, including Rett syndrome. Greenwood Genetics Center have been, during the 40 years or 40 plus years of its existence, very focused on developmental disabilities. And Rett syndrome, as well as other disorders, have been a focus of interest. And the way that we're going to do this is to bring the best experts everywhere. We're going to be focused on doing the best and faster development that we can, and whoever is available and willing to collaborate with us is going to be part of this endeavor. So I'm going to mention how this is going to affect the existing trial in a minute. The next one. Next slide. So let me give you a brief update. Given the time, I want to make sure that we have time for answering questions. When we're talking about the IGF-1 trial, we're talking about the one involving Increlex, which is the commercial name of this, the full-length IGF-1. This is the whole molecule, not only the head of the molecule, which is essentially the NC drug. And we're talking about two trials. One is the phase 1 trial. Remember, each trial is a discrete entity because it's a different goal, different population. It's not like a single trial that goes through multiple stages. In some ways that way, but in terms of the FDA and in terms of data analysis, each one is a separate project. So the phase 1 trial ended in July 2012. It was my first job when I came to Boston to complete the trial, which had been initiated by Dr. Omar Kwaya. And it was a small trial, like typically phase 1 trials are. And it was very important because, as you know, a phase 1 trial is needed to show safety. That's the main goal. Having said that, the design of the trial, we did a modification to the original design to be able to collect as much data on efficacy, on the effect of the effect of the drug on symptoms. And that's very important because the more that you know about the potential symptoms or aspects of the disease that the drug is targeting, improving, not only from the animal model work, which is important, to give you some general idea, but also from patients, you are going to design the best possible phase 2 trial. This is where the field is changing, with the idea of combining phase 1 and 2, maybe doing a couple of phase 1-2 trials to have a better idea of refining what are we going to target in terms of what is really what the drug is doing. We published this at the beginning of 2014, more than a year ago, and that has been the foundation and the sort of green light to go to phase 2. Now, the phase 2 trial was designed to be a phase 2 followed by a phase 3. And when you design a project, you design the project depending on what you want to do. And we wanted to go a safe way, a way that we will have at the end data will be interpretable, not taking big risks, but going a reasonably fast pace, but a process with clear goals. And the goal was to show that IGF-1 is efficacious, to really expand upon what we saw in phase 1, and eventually narrow down the situation and replicating the situation in phase 3. So we knew that phase 1, from the phase 1, that IGF-1 was potentially able to treat anxiety symptoms and breath holding. So we focused on that, but also we added other aspects. We didn't want to miss the chance to see improvements in any other neurologic symptom, motor function, communication, other type of intellectual function. You could imagine the whole range of aspects or symptoms that you can see, impairments that you can see in Rett syndrome. So it was designed in such a way that we had 8 primary outcome measures. So if one of the 8 was working, we could go to the phase 3, focusing only on that one. That was the idea. At the moment, I will give you a more complete update. We are all 30 subjects have been enrolled, and almost half of them have completed the trial. Now, unfortunately, we didn't go at the very beginning to the FDA, so it wasn't possible. There was a lot of excitement about the trial, so we needed to start right away. And so we went to the FDA early this year, and we got the positive feedback from the FDA. But because all the history of the trial and the focus also of the drug company that produces Increlex, we decided to focus on to narrow down from 8 primary outcome measures to a single one, which the FDA really likes, particularly for the phase 3 trial. But they were agreeable on that, but that meant that our power, remember what Dr. Kaminski said, numbers, numbers, and numbers. The FDA requires the numbers, so we need to expand the trial. And I'm going to tell you what the expansion is going to be. The next one. So before we go over that, let's review what we did. On the top of this cartoon is the phase 1, where we demonstrated the drug is safe, is well tolerated. The parents do a terrific job injecting this subcutaneously. We all like oral drugs, but parents can do a very good job with injectable drugs, and this has been the case, no complications. And we demonstrated that, as I said before, that the drugs seem to be efficacious in terms of anxiety and in terms of breath holding. And here you can see the design. And what we learned about what the drug could be doing in terms of symptoms during that 20-week period. Even though it was open label, we got data that we thought that was reasonably objective to plan a phase 2. And the phase 2 trial is a crossover design, long. Unfortunately, things happened, and the washout period had to be extended due to an issue about the manufacturing of the placebo that the drug company didn't provide. We need to go out there and to buy the placebo, which is not inexpensive to do that for multi-injection. So that 10-week period has almost triplicated, and that means that the trial is now very long. And we were able to get from our ethics committee approval to prescribe the drug, even if not everyone had finished the trial, to prescribe the drug if the family concluded that the drug had been beneficial, even though they are blind and we are blind to which half of the trial the girl was receiving the drug. So this is a trial for girls between 2 and 10, but they had to be, the young girl had to be in the post-regression period, very important, because if they are still losing a skill, as you can imagine, we're not going to be able to have a stability to see the effect of the drug. We are not going to know whether there was less improvement because they were still losing a skill no matter what, and the drug couldn't reverse that. So it had to be very important, it had to be stable at least 6 months without loss of skills. And why age 10 and why we don't go higher? Because this is a hormone, and not only is able to work on the insulin pathway, but also works on in growth, and is really a big player in the growth hormone pathway. And for that reason, there is a possibility of having negative effects on growth, meaning we can stop growth before it should stop. So, so for all those reasons, our colleagues in endocrinology recommended to be on the safe side, particularly knowing that a big proportion of girls with Rett syndrome have early puberty, that we better stop early, not at age 12, and that's the reason why we do it at age 10. The next one. So this is where we are. As you can see, we have done a lot of work. We have pre-screened a total of 63 girls to end up with the 30 that we need for the trial, and everyone has done at least visit 3. So at least everyone has been almost completed the first half. As you can see, the number of girls who have completed visit 5, the last of the first half, is almost the 30. So we are essentially finishing the second half, or in between. Some are in this very long washout period that unfortunately is long because of the issue of the production of the placebo. A number of them have finished the trial, 13 of them, but a second wave of about 6 to 7 is now pretty close to finishing the second half. So we are going to have almost 2/3 of the girls finish it in the next few weeks, definitely before the by the end of the summer. So, so it has been a safe trial as the phase 1. We have replicated that. Everyone who has exited the trial without any problems. So quite a few of the girls have decided, like you can see, 11 of the 13 have decided to stay on the drug, and it has been so far safe. The next one. Briefly, because we don't have a lot of time, I realize that I have spoken too long. I'm going to run through these slides. This is based on animal data, mainly from Rigan Kasur's laboratory, both the peptide as well as the full length IGF-1. The next one is for illustrating. These are the graphs showing the improvements in the scores on the questionnaires. For the anxiety measure, we also have data from EEG that supported this. I'm going to skip that. The next one. To mention the other key symptom, this is more objective data because we measure breath holding by this objective plethysmography. Essentially, we don't observe, but we measure in a very quantitative way breathing. The next one. And you can see that breath holding, whether breath holding was longer than 10 seconds or even shorter than that, both were improved in the phase 1. So we are hopeful that that's going to be found in the phase 2. The next one. So I want to finalize by the changes. So what does it mean? What does it mean that what FDA required to add more subjects because we narrowed down to one primary outcome measure? What does it mean that I am leaving Boston? First of all, that it means that we need to go from 30 to 47 subjects, at least 47. We can add more than 17 additional subjects, but that's the minimum required based on statistics. And we are going to be able to start these in the next few weeks. And we need to add other sites. Why is important that? Because what Dr. Percy mentioned is important because it's easy to do larger numbers if you distribute them among sites, but also because the FDA likes to have data from different sites to show that there is no bias. There is no systematic bias because people do the same thing in a particular way in the institution. And also, if we do something wrong here in Boston, the other two sites are going to help in saving the trial. So at the moment, Greenwood Genetics Center and Baylor have committed to being part of this. So that means, unfortunately, the trial is going to end by early 2017. Now, what happened with the transition of Boston Children's? There's not going to be a new PI at the main site. This will remain as the main site. And the person who has been designated is Dr. Mustafa Sahin, who has considerable experience in NDD trials, neurodevelopmental disorder trials, and he's an authority in tuberous sclerosis clinical trials. So he's quite experienced in running trials, and including those not only industry trials, but also trials like the one that we designed here. So he's familiar with doing that. In addition to that, Dr. David Urion, who is the head of behavioral neurology in our department, who is a clinician with considerable experience in seeing children with Rett syndrome, will be supporting the trial in terms of looking at the day-to-day operations, adverse events, and so on. I'm going to continue remaining involved in the trial in a sort of a supportive supervisory capacity and making sure that the trial is well implemented, that we communicate with FDA properly, as well as with the drug company. So I'm going to be coming back to Boston to examine some of the kids. I'm going to finish all the evaluation for the children that I have been following personally, and I'm going to remain involved. The next one. So simply to mention that, as I mentioned before, this is all a team endeavor, and here you see different pictures of members of the team and some of the participants. This has been a long journey, but a journey that has been successful thanks to everyone being involved, and thanks to all of you, the family who have participated. Thanks.
Thank you so much, Dr. Kaufman, and thank you, Dr. Percy and Dr. Kaminsky. You've given us a lot to think about and a lot to talk about, and I, for one, on behalf of all of our attendees today, thank you for all of the very rigorous and detailed and difficult work that you're plowing through. I know this is a tough ground for you, and we appreciate all of your efforts on behalf of our kids. So I just really wanted to get that out there. We have about 5 minutes left for the scheduled webinar. If everyone can stay on, I'd like to go for about 20 minutes if we can, because we have a number of questions in the queue. Our panelists, can you stay on the line for about 20 minutes?
Sure.
Great.
Sure.
Okay. Great. Dr. Percy?
Yep.
Wonderful. Okay, so we have a number of questions. If we don't get to all of them today, then we'll be sure to develop a question and answer document, and we will continue to have webinars of this kind. I think this has been a tremendous format for us, so this won't be the last time families will have a chance to hear from you. Thank you for joining us today. Okay, so if I can, we have a number of questions from families who perhaps didn't have a chance to attend our earlier webinars this week. Dr. Kaminsky, Steve, if you could begin at the beginning for us, can you just explain a little bit about what MECP2 is, who has it, who doesn't, and the possibility of how it's going to be used for a therapeutic intervention? Can family members with healthy MECP2 donate it to their kids? Is that something that's being looked at? I know we could spend an hour talking about it, but if you could begin at the beginning on that for us, that might help a lot of families.
Sure. First of all, every one of us has MECP2, and it's actually expressed during our development of our central nervous system, of our brain, and it's there every day in the cells, a very large protein, a gene coding for a very large protein that in many ways acts like a conductor in the symphony, which actually tells other proteins to be turned on or turned off, much like the conductor tells the first violins, the second violins, and the violas and the cellos to play when they're supposed to play. It is a very large protein that actually is in every cell. We cannot give it therapeutically because it could never actually get across the blood-brain barrier, go from your blood into your brain, and then get across the cell membrane from the blood into each of the different cells it needs to get into. So the MECP2 that's abnormal in Rett syndrome actually doesn't do what it's supposed to do. That conductor doesn't conduct the symphony in the right way. So actually giving MECP2 therapeutically would never work because you can't get it to where it needs to be in order for it to do what it has to do. So you have there's three options, but two are very viable. Can you fix the gene so it can make a normal protein? And many people are trying that. We're supporting some research on that. The NIH is supporting research on that. RSRT is supporting research on that. Can you use gene therapy to fix the gene? That is a long ways away from being actually something that could ever be used in a clinic. There's a lot to be done there, so that's way down the road. The other thing that you can do is, instead of trying to fix the conductor of the symphony, can you fix the players in the symphony and coordinate them to play when they're supposed to play without the conductor? And that's what both Alan talked about with 2566 and Walter talked about with IGF-1. If you can get other pathways to do what they're supposed to do when they're supposed to do it in the brain, you may be able to overcome the deficiency of MECP2. So can a parent donate a healthy MECP2? No. I mean, yes, they could, but it's not going to do anything because it never can be delivered to where it needs to do what it's supposed to do. We have to get it into the very, very internal spaces within the cell, within the nucleus, to do what it has to do. Hopefully that helps, Paige.
Great, thank you. If anyone has further questions on that, go ahead and send it into the queue, and we'll get that answered. Dr. Percy, we have a number of questions going back to your discussion about the next phase of the natural history study. Families that were enrolled in the previous natural history study wonder if they will be able to enroll in the new version, if you are really hoping that families that you've been following for many years will continue, or if you're only looking for new families.
Obviously, that's a great question. We want everyone who has been in the previous natural history study and who would like to continue. And that can happen in two ways. One, you can stay with your original enrolling site, or if distance is an issue, we are happy to transfer the controls to the site that you select. And once that protocol is approved, and we hear from parents, we will do that directly. So yes, we would like to have everyone who has been in the study and wishes to continue in the study to do so. As I said, we have enrolled 1,200. we don't have 1,200 in the study as yet because of some losses due to moving, some of the problems with the recession during the last part of the first decade of the 2000s, and so we need additional enrollees as well. Now, for example, if you were seen by us in Chicago and you lived in Minnesota, and assuming that the Gillette Center is brought online fairly quickly, we would be delighted to transfer you to Gillette, so your travel would be cut down, and so on for the many, many other families who would otherwise have to travel great distances to go to the original enrolling sites.
Thank you, Dr. Percy. And we have a number of families outside of the U.S. who are asking about enrollment, and this would probably generally apply both to the natural history study and clinical trials. Can you talk about non-U.S. citizens, how they would enroll, or if there are opportunities for, say, a natural history study going on in any other countries that you know about? Can you speak to those?
Because of the prohibition for the U.S. to provide money for sites in foreign countries, we cannot do that. If the foreign country can generate sufficient funds to support their site, and we get word of that, we can then make application to the NIH, the various offices there, as well as the Data Management and Coordinating Center to gain approval there. There is additional problem with enrolling sites outside of the U.S., perhaps not Canada or Mexico, but certainly from farther distant countries, in that everyone has to be audited by the DMCC usually on an annual basis, and the cost of conducting an international audit is quite high, and therefore that site would also have to support the costs of an audit. But yes, that we would not prohibit anyone from enrolling in any site. Now, I can say we do have a number of persons already enrolled from other countries who, for one reason or another, travel to this country, and the child is seen here. So we are open to everyone, but we have problems in funding sites from the U.S. That do not occur in the U.S.
If I can answer the issue about the clinical trials, we, in our trial, and in general, it's not recommended that we recruit children from other countries, and the issue is to keep track of safety data, and also to make sure that we are meeting the drug without serious adverse events. Safety, as we mentioned before, is a very critical issue, particularly in the early stages of clinical trials, phase 1 and phase 2. The infrastructure is required to do trials across countries is something that only big companies have, and there are other regulatory issues. For instance, that all documentation has to be in English, so we need to translate documents. Let's say a medical record into English, and that's actually very expensive because it has to be an official translator. It has legal implications. If the translation is not accurate, there are legal consequences. So it's not practical, it's not feasible to do trials across countries.
Right. Thank you. That's a very good explanation. Thanks, Dr. Kaufman. Dr. Percy, we have a number of questions about trofinetide in the NNZ trial. Parents have read that Neuron would be publishing, possibly Neuron, perhaps the PIs, would be able to tell which families were on the drug and which were on the placebo. Do you have a timeline for that? And also, do you have a timeline for when the next phase could possibly start?
Yes. So, in terms of letting families know whether their child or daughter was on placebo or control, we had a fairly lengthy. If anything that occurs in 1 hour can be described as lengthy, but we did have a discussion with the FDA about whether or not individuals who had been in the trial, whether they were on placebo or drug, would be eligible for future trials if they happen to know their drug assignment in this trial. So the company, which is in control of the data, is, I think, coming to a final conclusion. I believe that the information will be available for those who want to know it. But for those who do find out what assignment their daughters had, they would not be eligible for any further trials with NNZ. Not that they wouldn't be eligible for other trials, they would not be eligible for the Trofinetide or NNZ study. So that's a significant consideration.
Great. Thank you. And I know you'll review that with every family individually who participated. There is a question for families that have participated in the IGF-1 clinical trial. If the NNZ trial opens up in a younger age, could that be a potentially exclusionary criteria if they were in the IGF-1 trial, that they would be able to be included or excluded from NNZ? Can you comment on that at this time?
Actually, I don't know the answer to that. I suspect under the same circumstances, at least if they didn't know what their assignment was, they might be. But I think the fact that they had been in an IGF-1 trial might preclude them from enrollment. However, that question has not been addressed as far as I know specifically, and I didn't really get to the answer of when the next trial will take place. I would suspect it's at least 6 months away. The discussion with the FDA was about several trials going forward, including the necessary registration trial, which would be a phase 3. The company now is back in discussion amongst themselves, deciding exactly how they're going to conduct the next phase of the trial. The next phase of the trial would be in girls 5 to 15. But I think it would be premature to tell anything until those determinations have been made by the company.
Okay, we look forward to having more information revealed as progress is made, and we'll certainly invite you back for a webinar to talk to families when those details come out.
I might say that when the one side of it, when industry is in control, and they generate the study, they exercise, and rightly so, complete control over how the trial is initiated.
Okay. You know, there was another question about the natural history study that I want to get in before we go to our next question, because I think that'll be it for the natural history study. We've had a lot of families who joined a little bit late and are asking about their sons with Rett syndrome, MECP2 mutations, MECP2 duplications. Are they eligible to enroll in the natural history study?
Yes. We have the next, even in the old iteration, but in the next iteration of the trial, there are 3 separate segments. One is for individuals with Rett syndrome, whether classic or atypical. The second section is for individuals with MECP2 duplication, and the third section is for individuals who may have a mutation in MECP2 or in other genes, but do not have Rett syndrome. Nonetheless, they may have significant developmental issues. So those individuals would be warmly welcomed into our natural history study.
Great. I have some very specific questions about NNZ. I hope that you can answer these about trofinetide. Can you talk a little bit about the construction of the trial, why the dosing was developed at the level that it was developed at, why a 2-to-1 ratio instead of 1-to-1, and there was a question about hyperthyroidism and why that was an exclusionary criteria.
Okay, so the actual discussions about dosing, I think, were based in part on trying to extrapolate from the IV study on Army personnel to an oral formulation. And they wanted to go with a lowest dose, which they felt would be safe, but might produce a recognizable clinical response. And they also recognized that one dose wasn't probably adequate, so they went with a 2-dose scheme. Going forward, we know that the FDA has actually encouraged using much larger doses, so doses up to 100 or milligrams per kilogram per day, on a 2 times a day dose, and perhaps even higher are being considered. So I think the issue was simply to be within a range that was safe, number one, and number two, to offer 2 doses so that they would have at least 2 endpoints or 2 points of determination for that. And there was a second part to that question?
Yeah, do you have an idea? Can you also address the time span, why it was chosen only 28 days on drug? And then, go ahead.
That's a very typical period. I actually learned on Monday for the FDA for a safety, a phase 1 type trial. They generally do not go beyond 4 weeks. In fact, we actually discussed going up to 42 days with the young girls, as this drug has not been proved safe in younger individuals. So, and they were taking that under advisement. But that is the standard time period, 4 weeks.
And actually, it was also the same period of 4 weeks for the phase 1 safety pharmacokinetics part of the IGF-1 trial also.
Okay, thank you. I think we're going to do 2 more questions because both of these might require a little bit of time, and then we'll make sure, again, that all of these great questions that have come in the queue do get answered. One of them has to do with the structure of clinical trials. In general, do clinical trials need to go through all 3 phases before reaching market? And the second question that came in that is related is, in general, have clinical trials ever been abandoned for efficacy reasons as opposed to safety reasons? So if a trial is continuing through all the different phases, can one presume that you're seeing improvements if they are, even if you can't publish results, if we see them moving through all the phases?
Okay, let me answer those questions. As I mentioned in my talk, we're considering, particularly this comes from oncology, the possibility of not going through all 3 phases. But the reality is that phase 1 and 2 and 3 represent the goals of a trial, of a series of trials, or the process of drug development. You have to demonstrate safety, you have to demonstrate efficacy, and you need to be sure there is efficacy. If you can do that in an abbreviated fashion, it's fine, but you have to do it. So, and you need to be able to separate safety from efficacy. So, in theory, it's possible not to go through each one of these phases. And that's the reason why now a model that is not so uncommon is to do phase 1/2A and then to go to phase 2B, meaning that phase 2A is as a variable dose and the phase 2B alone is at the fixed dose. So there are different ways to do this, but it requires a lot of good planning, in the same way that the question about why the ratios are that particular way, because we want always to have more patients with the drug, but if we decrease the placebo group so much that we don't have statistical power, it defeats the purpose of comparing drug versus placebo. So the idea of making this more expedited is there, but how to do it is the challenge. Now, if you see a trial progressing through 1, 2, and into 3, you should assume that exactly that the drug had been safe and efficacious. The FDA, particularly these days with all the regulatory aspects and clinicaltrial.gov, there's not going to be IRB, even if the FDA is not involved, there's not going to be an ethics committee or IRB that are going to accept to move from one to another unless you have demonstrated that the goal has been established.
I can add that the general rule of the FDA is that there have to be 2 phase 3 level trials, independent, and the results have to be similar. But with rare diseases, they have approached that in a slightly different way, and with a very good single phase 3 trial in a rare disorder, they are willing to go for registration with that, recognizing the problems of numbers. Am I aware of trials that have gone to phase 3 and been interrupted? Yes, there have been some trials that have been stopped because the first or second phase 3 study did not substantiate the efficacy in the first or the second trial.
Yeah, I would like to add that the trials should have an independent body other than the FDA, making sure that the drug is the trial is progressing in a safe way. But that body called DSMB also can look at efficacy, and if in a trial, typically in a phase 2 or 3, if there is no evidence of efficacy, what is called futility, the trial can be stopped because nothing is happening, and let's say at the midpoint of the trial, there's no point of continuing. But the investigator usually don't know that because it's a blind study. It has to be this independent body, which is the DSMB. The other thing that can happen also if at that midpoint is demonstrated that the trial is so successful that really there's a big difference between those who are on the drug and the placebo, there is a possibility of stopping the trial for that reason. So there are a number of circumstances, but it has to be done in a way that is scientifically valid. There is also the very remote possibility that in a phase 2 trial, if it's extremely successful, becomes a phase 3 for a rare disease. The FDA will consider that, but there are many conditions that have to be met. It had to be a sort of wow effect, spectacular, to be able to skip the step from phase 2 to 3.
Yeah, that's what I call the penicillin effect.
Yeah.
You don't need much of a statistical analysis to prove this penicillin works.
Thank you. Thank you very much. I know this is it's really important for all of us to understand that why we have to be patient, why certain phases and requirements are in place. We're all anxious to have treatments get into the general clinic to be available to our kids, but we also really want to make sure that all the I's are dotted and T's are crossed for the safety of our kids, and also for when we get to the table and consider enrolling our children in a trial. I think it's really important that we all understand the work that's gone in before they reach our girls to make sure that you're really watching out for their safety. So we appreciate all of the information you've shared today. We have one more question. Perhaps Steve Kaminsky, you can address this one, and then we'll close out our webinar today. And this might take you a few minutes to answer. We have a lot of questions about other clinical trials that are happening beyond the 2 that we discussed today, both in the U.S. and around the world, and if any of those are in phase 3, but maybe some of the different phases. Steve, you gave a nice presentation earlier this spring that was comprehensive about all of the trials. Could you give us a brief update today, and we will do a more extensive webinar in the future reviewing all clinical trials that are underway.
Sure. So right now there's 7 clinical trials, 2 of which have been completed. You heard about one of them today, the NNZ-2566. That first phase 2A is complete, and again, more are planned. Another drug from Edison Pharmaceuticals called FE743 was a phase 1 trial in Italy And that particular drug is a drug that actually helps with oxidative metabolism, the ability to essentially really work the energy pathways in the cell. That particular drug was safe and tolerable in children, and there was a secondary outcome measure, head circumference, that looked very promising. Edison is thinking and planning about possibly doing a phase 2 trial here in the States, but we haven't had any update from them in the last sort of 3 months. I'm reaching out to Matt Klein, who's their chief clinical officer, to find out where they are with that. You heard about IGF-1 today from Dr. Kaufman. Another drug that's in trial in France, Despiramine. That trial is closed, but we haven't seen the results of that trial. And that particular drug was one of the first drugs used to reverse symptoms in the mouse model, but we haven't heard much on the results of that. So again, we don't know the outcome of that. We're waiting to hear about that. Another compound that is being tested out of Novartis in Switzerland is fingolimod, and that is essentially a compound that stimulates BDNF, and BDNF is in the pathway, the similar pathway that IGF-1 is and NNZ-2566. It helps form synapses, synaptogenesis, which is one of our major things we're trying to do. That trial is in a phase 1/2, still ongoing, and is due to close this year. The next trial that I remember is one that's at Hopkins with dextromethorphan. That trial's been going on for a long time, and that particular compound is very important in the NMDA pathway, and that actually just helps with cell-to-cell signaling. That trial is due to end this year as well, and so hopefully the data will come out from that. And then the final trial is with Copaxone, and there was a phase 1 trial in Italy and a phase 2 trial at Montefiore. And Copaxone is a drug that's used in a number of different disorders where there is challenges around behavior. Copaxone actually stimulates the production of BDNF as well. So we haven't seen the outcome of those trials either. There are a couple of other trials that are in planning phases that are not up on any public websites yet. That is ketamine, the follow-up of the David Katz study that he did with mice, and that will be done at the Cleveland Clinic and I think at Montefiore, as well as another drug in Europe that Serazotan, which had a very successful trial in mice out of a lab in John Bissett's lab in Julian Payton's lab at Bristol, University of Bristol, United Kingdom. So there's some other stuff going on out there. I listed 7 that have been on clinicaltrials.gov. The 2 that I have listed that aren't on clinicaltrials.gov is Serazotan and the ketamine. And hopefully those will come up sometime in the near future. So you can see some of these trials have been going on for some time. You heard from Dr. Kaufman, with his extension that accommodates some of the stuff that the FDA said. He will be working through 16 and hopefully have that finished in early 17. So these take time. There's a lot of patients involved. Patients, i.e., we have to be patient. And the challenge for us all is we want to go quicker, faster, better, but to do it right and to do it with approval by the FDA, we have to make sure we dot every I and cross every T, because we do not want the FDA throwing a penalty flag at us and telling us to start again. That's the last thing we want, because of the resources we have to invest and the time we have to invest, and most importantly, the effort that the families have to put in and the things that our girls and children and boys and adults have to go through in one of these clinical trials. It is not trivial, and it is not for the faint of heart. I'll stop there, Paige.
Thank you very much. Okay, so we've gone a bit over. I think it was valuable that we did so, and thank you for staying on the line and fielding all of these questions. Like I said, this is a tremendous format for us to get information out to our community. Thank you for joining us today. Dr. Kaufman, good luck with your move. We're actually very excited about it, and thank you for reassuring our families that the work will continue, and this is a good thing for Rett syndrome. Dr. Percy, we wish you best of luck, and thank you for all the meetings that you've been in with the FDA and advocating for our girls always, our girls, women, boys. Dr. Kaminsky, thank you so much for all the work that you're doing on behalf of RettSyndrome.org to manage critical donations that are coming in to support the research that's on our full 360-degree spectrum of research. We can't abandon any area, and we're really encouraged by having this technology to bring this information to you. To all the attendees who stayed on, we appreciate your length of time for your screen time, and we hope you would join us for our next webinar, which will be tonight, this afternoon, with great information on overall quality of life. We can't forget today, although we're really, really encouraged by the research that is coming. We hope that you also learn from RettSyndrome.org the most current practices for keeping our girls healthy today. So we'll be looking at quality of life this evening, and we have a new webinar just scheduled for next week on occupational therapy. Take a look at our website, rettsyndrome.org, under For Families, and you can register for that webinar, which will be next week. We'll see you again next year in person. You know, technology is great for getting information out quick, but nothing can underscore the importance of families getting together to meet with our experts privately, publicly, and for families to get together one-on-one to share experiences and create friendship, bonding, and support. So we look forward to you joining us perhaps in Orlando in October, and absolutely plan to meet up with us in the Chicago area in June next summer. Thank you again so much. This now concludes our webinar for this afternoon, and we wish everyone to enjoy your summer or your winter, depending on where you are in the world. Thank you so much. Bye-bye.